Welcome to Life in Biology. I'm Dr. Joel Graff, and the last two episodes of Life in Biology have been about potential molecules that could stop the replication of coronavirus. This video is not coronavirus related unless you want to say that moving classes online because of coronavirus is a good reason to make some educational YouTube videos. I guess we're related there. In my class, we have just talked about cell signaling from receptors and getting a response in the cell, and we had moved into talking about cell cycle, and the next topic is the regulation of the cell cycle, and so that's what the next few videos will be about. So, quick update if you weren't in my class about the cell cycle. I have a number of green arrows drawn in a circle to represent the cell cycle. There's a start point right here that I'm labeling right after mitosis. So mitosis is when one cell splits to two cell. So we want to see how you go from having one cell and going all the way through the cycle to getting to two cells. So we're going to start there, and the first phase of the cell cycle that the cell would go through would be G1 phase, and then the second phase would be S phase. That stands for synthesis, and it's where the DNA is replicated. We have another G phase, G2. Now the G's stand for either gap or growth, depending on who's telling you what they stand for. And then M, the final phase of the cell cycle is mitosis, and there's lots of smaller steps within mitosis, but that's not necessarily important to this video. When you're going to regulate the cell cycle, you might want to do it at critical points, and so I have one checkpoint drawn here at the end of G1, a second one drawn here with two asterisks at the end of G2, and there are three asterisks crammed in there for the third checkpoint in the cell cycle, and that's sometimes called the M checkpoint. Occasionally you'll see some figures where they have a checkpoint within the S phase, but we're leaving that out for this discussion. All right, so G1, at the G1 checkpoint, this is a point in which the cell commits to going through the cell cycle. So the cell would want to make sure that there are nutrients around. Then the cell needs to get some signals from growth factors, and that's what the focus of the second half of this video is going to be about. DNA damage. If you have any DNA damage in the cell at the G1 phase, you want to get that fixed before you go to S and copy the genome. And then finally, cell size. I was taught that G meant gap phase, but I've seen it growth phase before. So this is a phase where the cell grows. If you're going to split into two cells, the cell needs to grow at some point, and that's what we're giving to those G points, or G phases. At the G2 checkpoint, right up here at the end of the G2 phase, you also want to check for DNA damage. Specifically, did the DNA get replicated properly during the S phase? And then you want to, again, look for cell growth. Are we big enough now for that cell to break into two daughter cells? And then finally, the third checkpoint is the M checkpoint, and it is right in the middle of mitosis. M does not stand for mitosis, but instead it stands for metaphase. And that's the phase where the sister chromatids of each chromosome are lined up down the equator of the cell, and the cell checks to make sure that things are the ropes that they're going to use to pull on those chromosomes to pull them apart into each new daughter cell, make sure they're connected from both sides. Okay, so like I said, this video is going to focus on growth factors, and that brings up the topic of cyclins. Before we get to cyclins, though, I want to mention that there's a counterpart to cyclins called cyclin-dependent kinases, or CDKs. CDKs are generally available in the cell, and it's only when cyclins become available do the CDKs become active. Now, cyclin-dependent kinase, by that last word kinase, we know that we're dealing with an enzyme that will phosphorylate other enzymes. It looks like I have two of the same picture drawn here, and I just wanted to try to help people avoid confusion. If you look for cyclins with a Google search, you're going to get a couple hits under images. The first one I think will probably be from Wikipedia, and the second one will probably be from Khan Academy. Khan Academy gets their figures from OpenStax sometimes, so that's where this first figure kind of came from. I'm copying it without asking for if that's okay. Don't tell them. And then on Wikipedia, the figure looks like this. Now I've color coded it, and you can see that the colored loops are the same. And so what we have here, as you go from the start right here through G1, over to S phase, over to G2, and then through M, you have the cell cycle just drawn out in a straight line versus a circle like we had it on the previous page. But that last step is mitosis, and at the end of that you get two daughter cells. Okay, and then the y-axis, I don't have it labeled, but it's just the abundance of whichever cyclin you're talking about. For example, this first red line says G1 cyclin, and it becomes abundant during G1 phase, holds out, and then during mitosis the concentration goes down because the cell has split. The next cyclin is called G1/S. The next one is, the one after that is called S cyclin, and then the final one is called M cyclin, or at least that's the terms they use on the Khan Academy and OpenStax. These terms, while they make sense and are easier to kind of remember which ones are upregulated at what time, especially if you can remember that the cell cycle goes G1S, G2M. Historically, the names of the cyclins have followed more like what the Wikipedia image, and this is, we're currently in March 2020 to date this, but currently that's what you'd see if you went to Wikipedia. Okay, but up here, G1 cyclin is called cyclin D. G1S is called cyclin E. The blue line represents cyclin A here, and the purplish pink line represents cyclin B. So D, E, A, B is the order of the cyclins there. So whenever these cyclins become abundant, no matter what you call them, they're going to bind to a particular cyclin-dependent kinase. There's different cyclin-dependent kinases, and each one will bind to a different cyclin. So when this cyclin D becomes, or let's call it cyclin G1, when that becomes abundant and the G1S becomes abundant, those will bind to specific cyclin-dependent kinases, and then there's going to be a specific set of targets in the cell, usually proteins, that are going to be phosphorylated by this cyclin-dependent kinase complex. So those two proteins work together to go phosphorylate things. So I mentioned earlier that the G1 checkpoint commits the cell to the cell cycle. So it's the checkpoint of no return. In the first step, you need some growth factors around, and they're going to bind to receptors on the plasma membrane of the cell. And when that happens, I'm leaving out a lot of detail, but there's a cell signaling pathway that leads in the end to the upregulation of cyclins D and E. Okay, those are the first two cyclins if you use the Wikipedia nomenclature. Once you have cyclin-dependent kinases forming complexes with cyclin D and cyclin E, then you get phosphorylated targets. Phospho is my shorthand for phosphorylated, and then RB is the shorthand for the name of a protein called retinoblastoma. So RB is much easier than saying retinoblastoma. If the cyclins and the cyclin-dependent kinases are causing retinoblastoma to be phosphorylated, what you have is that the RNA or the RB protein has a lot of phosphate groups on it, so it's hyperphosphorylated. Hyper means a lot of phosphate groups added to it. And then there's a second protein I brought into the picture here, E2F. E2F is a transcription factor, and when RB is phosphorylated, E2F hangs out by itself at a promoter of a bunch of different genes. These are S-phase genes, and when it's sitting there, transcription machinery comes in and mRNAs start to be made of the different S-phase genes. In contrast, if you do not have an active cyclin-dependent kinases because they're bound with the cyclins, you will end up with an RB protein that only has a low level of phosphorylation events. So I drew 1 phosphate here versus 5, and that might not be the actual numbers, but the idea is only a few versus a lot. When you have hypophosphorylation, low level of phosphorylation, the RB will bind to that E2F transcription factor, and then you do not get transcription. So this arrow up and over, that green arrow here, it didn't have a red X over here, which means that these genes were being made. It does have a red X here, and that means that the retinoblastoma protein, the hypophosphorylated form, is doing a good job of preventing E2F from getting these S-phase genes turned on. So going back to our original slide here, whether you can get past this first checkpoint from G1 and move into the S phase, you need the RB protein to be hyperphosphorylated, and that can occur when cyclins become abundant and bind to the cyclin-dependent kinases and cause the phosphorylation and release of RB from the transcription factor that turns on the S-phase gene. So maybe you'll need to watch this video a couple of times because I feel like that was a ton of information, but hope this helps anyone that's out there struggling with the G1 checkpoint regulation. All right, like and subscribe or not.